So we are very lucky to have Maxime Taverne from Hôpital Necker, who will introduce to us, I think, the real-life aspects of morphometry and its applications. Thanks a lot, Maxime.
Thank you, Jean, for the invitation. Hello again, everyone. I'm working as a postdoc fellow in Hôpital Necker and Institut Imagine in Paris. And since a year or two, I'm also co-head of the Craniofacial Growth and Form Lab. It's a brand new lab that was created within Necker Hospital by a maxillofacial surgeon to address, as Jean said, real-life questions as, what do I want to know as a clinician, as a surgeon, to better manage my patients. And this lab is focused on describing and explaining the mechanisms behind craniofacial growth. And so I will show you the different aspects of our work. And also to start with, I'm an evolutionary biologist. I did my PhD at the Museum of Natural History in Paris. And in this regard, my background in the study of shape is more applied background. So I know a bit of theory, of course, because I want to understand what I'm doing. But let's say I'm more of a motivated and passionate user of morphometrics rather than, so I'm not a mathematician. So in advance, I apologize if some of the technical aspects I'm going to present are maybe, if you find small errors. What we study in Necker Hospital. So Necker Hospital is the oldest pediatric hospital in Paris and in the world. And we are highly specialized in the management of craniosynostosis. So craniosynostosis is a range of diseases that can be either genetically based or not, and that are characterized by craniofacial malformation due to early suture closure of the skull. And these malformations, you can see that they are characterized by, they're characterized by here early fusion of one of the coronal sutures, or both coronal, or the interfrontal suture. And you see that the constraint of growth is compensated by some additional growth elsewhere. So where the brain still needs to grow, still needs to find the space to develop. And so there will be compensatory growth that lead to the malformation. And so these craniosynostosis could be idiopathic. So it means that we haven't found any genetic basis. So it could be just a spontaneous developmental disorder, or also epigenetic. For example, it is quite usual to have what we call positional plagiocephaly. So let's say a kid has a neck pain, or is not really active, when he's a baby. So he's always sleeping on the same side, And so the skull is flattening on this side where he's sleeping. So it needs a correction, not necessarily surgical, but yeah. So craniosynostosis, it ranges from this positional mild, very mild condition to more severe conditions involving these single suture craniosynostosis, and to even more severe phenotypes. And in the case, it's the case of syndromic craniosynostosis. So these diseases are related to mutations in a family of gene, which is encoding for some receptors that are overactivated. And so the bone is forming from, membranous and also cartilaginous tissue. And this ossification rate is too high. So it is a gain of function mutation that overactivates these receptors, and eventually the bone is forming too fast and too soon. And so multiple sutures or multiple growth sites in the skull will be ossified too soon. and so the brain cannot grow normally. So the main syndrome that we study is the Crouzon syndrome. It's the most common in this family. And so you have here a very diverse presentation of patients from rather mild to very severe. And it's a condition that, if not treated, it can lead to death of the patients very, very young. So these patients, they need a very complex management, including lots of surgeries throughout their growth. From very young, in these Crouzon patients, first surgeries can happen before the age of 1. So first thing that surgeons do is this kind of procedure. So it's a posterior expansion of the skull. So it's cutting the back of the skull and putting some, distracting the posterior fossa. There are also lots of different kind of surgeries that will be eventually done to increase the volume of the skull to allow the brain to expand. And also, of course, there are some severe. I want to go back. Yes, there are also some severe malformation of the face, and the orbits can be too narrow, so the eyes are just popping out, etc. So there are lots of ophthalmological complications, and so at some point they also need what we call monoblock procedures. It's really, really invasive procedures. 1% death rate during the operation for these patients. It's really a huge operation, but very necessary in the worst cases, because it is the only way to expand the face, allow the frontal expansion, allow the enough space for the eyes, and also for the upper airway, so that they can breathe also normally. So it can be, yes?
And to be clear, in those slides, you mean that basically the same patient is going to have maybe 5 surgeries? Some
Crouzon or Apert patients, they can have 10 to 15 surgeries. And the problem is that it's a genetic disease that affects bone formation process. So as long as there is bone formation in your life, you're going to have problems.
Okay. And you also have problems in the limbs?
Yes, so in these mutations, there are also some patients, and especially in Apert syndrome, they have syndactyly, for example. So fusion of the fingers or fusion of the long bones. So for example, they can have fusion of the radius ulna, so they cannot rotate the wrists. Lots of other complications that can affect the whole skeleton. But the major concern, of course, for survival of the patient is the craniofacial region. And as I just showed you, the range of severity is really wide. And also for the same mutation, we have very different phenotypes, very different personal trajectories. And so for the same mutation, sometimes you're going to have these procedures, during the child growth, or you can have different trajectories possible. And this is really due to the inter-individual variability that is really, really high. So one of the main goals of our team is to better understand how the skull grows in these children, and also understand why it is so variable, what is causing this high variability between the patients, and how can we anticipate it. The idea is that, let's try to minimize the number of surgeries, optimize the surgeries when we can, etc. So that's the main motivation. And so to address this question, we try to have a holistic approach on the craniofacial region. We have projects on the brain function and anatomy that we are doing with Neurospin in CEA Saclay. Also, we are looking at the space available for the brain to grow. So the endocranial volume. We also study the shape of all the bones in the skull and also the mandible to understand the relationship between the anatomy and the function, protection of the brain, function masticatory, function breathing, function protecting the eyes, etc. So it's like morphofunctional approaches, like relating the morphology and what the organ is supposed to achieve. So yes, we have projects on the mandible and also on the upper cervical spine, because huge malformations can happen there also, and in relation with the central nervous system in general. And also the upper airway, as I said, because these malformations also they make the upper airway very tight, and some children they just can't breathe normally, or they can't breathe during their sleep. So the idea with all these combining all these compartments of the craniofacial system is to have something comprehensive for which we can have data for all the patients, and then try to better understand also the interaction between all these little units. So the approaches that we use in the lab we try to cover from the diagnosis to designing better therapeutic strategy. So first method, we use a part of the team is dedicated to use machine learning and deep learning approaches to make early diagnosis based on 2D and 3D pictures. So in Necker Hospital, we have a huge database of more than 1 million standardized 2D pictures for the last 45 years in the maxillofacial department, because we have this professional photograph taking pictures of every patient, frontal profile pictures in a standardized way every time they come to consultation. So huge database. And there was this PhD project done by Quentin Agnoc. He's a maxillofacial surgeon, and it was very, very fruitful, because now this part of the team is a foundation. It's called ID, artificial intelligence for dysmorphology, and now it's a foundation, non-profitable foundation, and the idea is to develop apps that can help either parents or clinicians to take a picture of the kid and have a diagnosis. And the idea is to develop these algorithms, to expand these algorithms to any kind of craniofacial malformation. So I think now they have included 40 different syndromes. So it's really working pretty well. Oh, this one is not going well. Mm-hmm. It's open office improvisation. Okay. Another part of our work is to try to describe statistically the growth of the patients. And so for that, we take advantage of all the CT scans and MRIs and pictures that we have to try to build these statistical form trajectories for the patients. Also, since we are a team that is in Necker, and also we are lab Imagine, Institut Imagine. I don't know if you heard about Imagine. Yeah, it's a foundation. It's a big research center for rare genetic diseases. So it's within the Necker campus. So there are Necker clinicians working in Imagine full time. Like the kids are coming to consultation for their genetic consultation in Imagine, and so there is a very good collaboration between the two. And some of their teams are specialized in animal model and cellular biology, etc. So what we do with them is some projects about the cellular structure of the sutures and the cartilage growth centers. so that we can better understand how the mutations affect these sites, and also what's going on in these growth sites, and how we can use this data to implement our predictive models. So speaking of predictive models, what our approach is biomechanical models, basically finite element models. And what we try to do is to design a numerical twin. And with this kind of model, we can ask some hypotheses about the growth, or how the sutures and this suture affect the craniofacial growth, and what are the forces involved, and what has the skull, the brain to do with this growth, etc. This is the kind of questions that we can ask. And in the end, so at the other side of the range of our projects, there is also developing some tools for spreading knowledge, and for also surgeons to practice the different surgeries. So we have had different projects on developing immersive video, and also developing surgical surgical simulators with multi-material 3D printing, etc. So we try to cover the whole thing. Okay, and so that's where geometric morphometrics comes into play. So as I told you, I'm from the evolutionary biology side of biology. In my field, my previous field, let's say, geometric morphometrics is landmark based. So you place homologous points on an object, and then you compare the 3D coordinates of these points. And it's really originating from paleoanthropology and paleontology, etc. So it's really like the traditional way of doing geometric morphometrics. And so the advantage of 3D GM is that you can run statistical tests and still preserving the shapes of the objects. The most important thing is that at the end of statistical analysis in 3D GM, the answer is a shape. So if you want an answer of how is this part growing, or how this anatomical part is affected by this drug, or is affected by this environmental factor, etc., the answer is a shape. So main component of shape variation with age is a shape. How, for example, apnea index is related to the shape, the answer is a shape. So it really helps you to visualize the influence of different parameters on the shape.
So what you describe now is the modern approach compared to as opposed to the landmark-based approach?
No, it's really the landmark-based.
Okay, so when you speak about shape, it's just the set of landmarks, or
yes,
the mesh?
Yes, the set of landmarks.
Okay.
Yeah.
And all the landmarks that you have to manually point on the shape, like the one you display on the screen?
Yeah, I'm gonna explain that. So quick recap, this side of shape analysis originated mostly with Darcy Thompson, that had the intuition that the diversity of shapes in nature could be summarized by these deformation grids, and one organ or one organism could be deformed following these deformation grids to end up to another organism's shape. And so it's quite how to say, so it was almost 1 century ago, because it started quite earlier than this. And then it was not formally developed. It was just an intuition, and it took other researchers at the end of the 20th century. So the main author in this field is Fred Buchstein. He was working in he's working in Pittsburgh and also in the University of Vienna. So that's where I went last year when I went to Vienna. And he really set up the theoretical field for the study of shapes based on landmarks, and also the theory behind the modern form of deformation grids, which are called thin plate spline. And basically, the latest developments of these methods were about end of 20th century, beginning of 21st. Really, the basic concepts, and this community of researchers are really developing packages, libraries in R. So it's really in R where you can find all the tools that you need to do landmark-based geometric morphometrics. And so that's where I will be talking about math without being a mathematician. So two main concepts are at the core of landmark-based GM. First, Riemannian spaces, and on the other one, thin plate spline. So the Riemannian space, so in the shape analysis on the biological side, it was formalized by Kendall at the beginning of the 60s. And so if you want to represent the whole range of possible shapes of a triangle, described by the coordinates, the 2D coordinates of their summits, the space is a sphere. It has 3 dimensions. But the thing is, when you have objects with higher dimensions, let's say 3D coordinates instead of 2D, and you have a higher number of landmarks, then this shape becomes multidimensional and nonlinear. And so one way to approach this and to use classical linear methods that we want to use, for example, in statistics to address research questions, then we need to approach this manifold space. And what we need is a projection in the tangent space here. So the shapes that are distributed in this field, we will have to project them in the tangent shape so that we can use linear methods in this Euclidean space. And this is possible in biological shapes because with a high number of landmarks, for example, the manifold field is really highly dimensional, but the variability of shapes that we actually observe in nature is rather small compared to this variability. So if we point where the shapes are actually located, it's reliable, it's relevant to have this tangent projection because the distribution in the whole possible space is rather small. So we need this. And so now we are in a linear space. And what is useful in the Riemannian field is that you can resolve some shortest path problems, so some geodesic, and you can say, okay, now I have this relationship between 2 objects, and this shortest path, this deformation, will be what I'm interested in. And the second aspect that is mostly interesting in, at the core of 3D GM, landmark-based, is the thin plate spline. So thin plate spline is some kind of metaphor to account for a deformation field between 2 objects that are characterized by 2D or 3D coordinates of landmarks. And the idea is that if you project a deformation grid on one object and you want to achieve deformation to reach a target shape, then you will have to deform this grid. And this grid will deform according to several parameters of optimization. And this optimization will be bending energy. So the deformation will make sure that the grid will remain smooth, and it will allow to make some interpolation in the shapes between landmarks. So limitation of landmarks, of course, is that it's punctual points that are on the structure. So what happens between these points? If we do not describe them, then we have some kind of missing data. The thin plate spline allows you to make some inference of the shape between these points. So it's an interpolation method. So it's Buchtin, and at the end of the 20th century, and then Mitreker and Dryden. So these authors, they tried to implement this thin plate spline also on what we call semi-landmarks. So it's really something that is currently used nowadays, not only landmarks, but what we call semi-landmarks. Semi-landmarks are constructed landmarks. They are not homologous. So basically, in biology, when you put a landmark on different specimens, what you are actually doing is putting a hypothesis of homology. You are saying this structure is the same in all the individuals. It's sometimes not so trivial, because if you're at the interspecific scale, for example, at a higher taxonomic scale, then sometimes you have very different species, not the same body organization. So it's quite a challenge sometimes to find the landmark that says, okay, this is the same structure. And for that, you have to make some hypothesis that comes from evolutionary biology. I mean, here we are working on the human skulls, so it's easier. But still, sometimes there are some parts that are fused, and you're missing the landmark, for example, or you have surfaces that are so smooth that you cannot find a reliable landmark. So the most typical example is on the skull vault. So easy to place a landmark at the junction between different bones, but then if you are on the very flat bone, what kind of shape descriptors can you have? And so that's the advantage of the semi-landmarks, is that they can be either placed on curves or 3D curves or 3D surfaces, and there will be constructed landmarks. So instead of being a true homology, there will be more or less a good approximate based on different optimization parameters. And the TPS or thin plate spline are playing an important role here, because they will allow to distribute the semi-landmarks on the surface. So yeah, I will answer your question a bit later also, but the semi-landmarks, they can be either first placed manually, but then they will be automatically distributed. Yeah, so to be a bit clearer, I will now take you through the typical pipeline of what we do to place landmarks and place semi-landmarks to have this high density shape description until the final analysis. Okay, so first thing to do, of course, that we need to do is have a mesh to work on. So this comes from segmentation of CT scans, MRIs, 3D photogrammetry, could be anything. Then we need to think, okay, what what kind of landmark do we want to use? So again, it's a hypothesis of homology. We have to think ahead of what kind of real anatomical homologous landmarks we're going to place. And then if we want to make the annotation of semi-landmarks at least semi-automatical, we need to prepare a template. This template can be either a very simple shape like this, or it can be one of the shape of the data set. It's possible. But basically, you're placing the landmarks in red, just as you would be doing on the actual shapes. And then you're placing also the curves manually, and also all the blue dots that would be the surface semi-landmarks. You're putting them, but only once on the template.
Okay, so to be clear,
Yeah.
you know, whenever you showed me those pictures,
Mm-hmm.
I assume that it was more like a patch. So you had
Yes,
your
it's a-
software
Yes.
to say, okay, cover with dots this area.
Yeah.
But actually, you perform the click for every single blue point.
I mean, you have different software solutions with patches. The only thing with the current solutions with patching is that it's more or less constrained. You know, in the software, like you can only, for example, delineate a region with 4 points. So it's rather limited in terms of application. If you have more complex bone shapes you want to manage, I mean, this is super quick. I do that in 2 minutes. You know, it's so you're just doing once. There are also other solutions right now in 3D GM, but this is, I would say, the most used and also the most flexible.
And also, like, because here you have colored the landmarks depending on their dimension. So
Mhm.
like in red, it's dimension 0, yellow dimension 1, and blue dimension 2. But I assume that in your software, you have a way of saying that this landmark is landmark 0, landmark 1, landmark 2, landmark 3.
Yeah.
And probably for the curves, you also have the indices. So I assume that actually in your software, like if you wanted to, you could visualize maybe a first blue patch in green, a second blue patch
Exactly.
in purple.
Yes, exactly. Yeah, you can name every landmark. You can, yeah. But anyway, there will be always in the exact same order, etc. So you can know that each landmark is corresponding to the same in the other specimen. Yeah. So if you do that once, then the next step is to, of course, place the landmarks and the curves on manually on the data set. And then you're gonna do the patching procedure. So everything is done in R. So basically what it's gonna do is there will be first rigid alignment between the template and the first specimen in the database. And then using thin plate spline algorithm, the template will progressively deform so that the landmarks, red and yellow, will slowly align with the target shape. And when there is approximately the same overall shape, then the blue points, the patches, are transferred on the target surface. So that's what you see here with all the little arrows. All the little blue points have been aligned, have been projected on the target surface. And so it's an iterative process and automatic. So after that, it comes to the next specimen, etc. So in the end, there is a projection of all the blue points of the template onto the surface of every specimen.
Okay, but to perform that, you need to click at least the real landmarks, at least the red dots on the specimen.
Yes.
So
Yes, there is a manual annotation for the rest of the data set. For the data set,
So
yes.
for the two first types of points,
Yes.
not the last one. The last one is
Yeah. So you manually annotate the curves with
Yes.
the dots,
Yes.
and for the surfaces, you hope for the best with.
No, you don't hope for the best because you can play with the parameters. You can also check any patching, but all the patching for all the specimens. Sometimes there are some little failures, so you need to adjust a little bit. But the most important part is then the sliding, because once the semi-landmarks of surfaces have been transferred onto the target shape, then you need to make them geometric analogous. You know, they are not anatomical homologous, but they should be optimized in a way that you can reasonably think that they are distributed in a non-biased way. Okay, so once they are projected, they will undergo iterative sliding and relaxing procedures, meaning that all the points will slide. So there will be some, inflation, deflation. So the blue points, they will be projected on the surface after several steps of inflation, deflation, according to their normals. So they can go back from the target surface and project on it. It just makes sure that at some point, some blue points do not fall on the other side of the mesh or whatever. And then these blue points, they will be slid according to their tangent on the surface. And in the end, the optimization criterion is that the bending energy is minimized. Yes. So in the end, the patches on one bone, for example, is really homogeneously distributed on the surface, and we reasonably assess that there are good estimates of the shape. Of course, we never compare one sliding landmarks with another sliding landmarks, because we are never sure of their really homology, but we can reasonably study the whole shape of the whole bone, for example.
And also because you say you do that in R, but there's
Yes.
a lot of human interaction. So basically, realistically, you have like your R studio,
Yeah.
you type stuff, and then you have pop-up windows that appear, and then you click, and then you close them, and then you go to the next one. Like, how does the graphical user interaction happen?
So in terms of code, there is the only real human interaction for this whole patching, sliding, etc. is really when the patching was done. We have this little toolbox that automatically opens a pop-up window. You just check, and if it's good, you go to the next, and so you check all the specimens because they have to be perfect. And if there is an error, then you have to maybe adjust something. There is a little human intervention.
Like, two kinds of workflow. Either your program opens pop-up windows, or it's more that you have a standard software where you do the annotation, that you export it as a CSV file, and then you load the CSV file from R. And I was just wondering, you're more doing the first approach,
yes,
instead of the second one?
Yes.
Okay.
But there are lots of different solutions, and there is also this not open access but still quite nice software. It's called Viewbox, and they allow you to place landmarks, curves, patches, do the sliding. But if you want to do the sliding properly, you have to pay for the license. So it's not open access. And you can run basic statistical analysis afterwards. So that's something that I found clinicians appeal to use because it's more or less everything in one software. So, but for me, I was just to code everything. Okay, so once we have all these semi-landmarks slid, then we can export everything as a CSV or TXT.
A last question.
Yes.
I work with people who do clinical trials,
Mm-hmm.
and they are insanely conservative in terms of software. So, for instance, like obviously they all use the same statistical models,
Mm-hmm.
but for some types of analysis, you have to use a specific software with a specific license. So it's not like you can come up with your implementation of the Cox model. You have to use a very specific version of it. Otherwise, they say, no, we cannot trust this. But for you, in your field, it's okay. Like you say, you're doing thin plate spline with semi-landmarks, and people believe that you have a non-buggy implementation.
Yeah.
Oh, okay.
Yeah.
Okay, no. That's good. I mean, for clinical trials, no.
Yeah, but if you want to develop a medical device for clinical trial, you like every tool that you use, everything should be like CE marked or approved at some point. So even for the CAD softwares, if you want to use CAD to create an implant, for surgery or things that are 3D printed, you have to use specific softwares and specific codes that were approved.
But for your
So
papers, it's not a problem, basically.
No,
Okay.
no. But it's a good question. Because that's if the project that we were talking about a bit earlier on the frontal curvature, I haven't really been in touch with people that could inform me on at some point how to really implement this as a and how to validate the code for a medical device. I mean, we have technology transfer and department in Imagine, and so.
Okay.
Yeah.
We had great training with Robin on this. People explaining us all the things.
I really.
Yeah, it can be done in an interesting way.
Yeah.
Okay. Yes.
Good stuff. Yeah.
Okay, and so after we have all the coordinates, we can proceed with mainly procrest alignment, so that we put aside any bias of shape, the orientation of the objects in space, and proceed with statistical analysis. And so, this is an illustration of a 3D thin plate spline, just to show how it can help us to produce morphology, 3D shape that makes sense in terms of statistical result. So as I told you, the very core theoretical aspects like candle shape, thin plate spline are already there for quite a long time, and to my knowledge, there was no major breakthrough in this community, in this field, for let's say 15 years. The major current development are to have this very high density shape characterization with lots of different patches. They are quite useful because in terms of, evolutionary question, it's really important to have a strong homology hypothesis in what you're describing. So if there are points placed on every bone separately, then you can really follow evolutionary trajectories of each bone. For the growth also, we really need this strong homology and characterization for all the bones. So that's it. The current idea is to make this more and more automatic. But it takes time to develop methods that will automatically place landmarks either on meshes or on 3D image acquisition like CT scans or MRIs. And it's because usually it's deep learning based, and so you're really dependent on the database that you use. And so if you want to make another automatic landmark placement, then you have to start from scratch, basically. So there are more and more people working on this automatic detection of landmarks based on CT scans. So this is an example on rats, but we have also started it in the lab on babies' CT scans on the head, but it takes time. But once we have this kind of tools to automatically place the landmarks, then all the rest of the methods of the approach that I presented, like all the semi-landmarks, since the process is already more or less semi-automatic, that would be a great help to have this automatic landmark detection. But it's not super reliable so far. The best results in cranofacial, at least, is based on surfaces, or if you have at least some if you have at least manual labeling, then it's easier. For example, if you have a skull and you have just a few landmarks to say, okay, this is the parietal bone, this is the frontal bone, this is it, then some people are now able to do some automatic shape characterization based on these labels. There is this group in the University of Colorado, and they just released, I think, 1 year ago, something like this, diffeomorphism model of the skull, and so of the skull vault, at least. And just by placing 4 landmarks on the skull base, they identify the skull vault, they identify the sutures, and so they label the bones. And once they have labeled the bones, it's easier to have some individual diffeomorphism model for each bone. So it's pretty neat, actually.
Okay, and just for instance, if you look at the skull on the right corner, how much human interaction was needed there? Like
for the face?
No, the one on the right, like the rat or something.
So
it seems to be very segmented into the different bones.
So on the upper right?
Yes.
So it's a salamander.
Okay.
It's still quite a lot of human interaction because you need to place the anatomical landmarks and then the curves.
Okay.
For all the bones.
Okay.
So it can, I don't know.
So it's still far from being automated.
Sorry?
It's still far from being automated for those kind of...
Yeah, as long as you have very, very diverse shapes, it becomes more and more difficult. For this example, yes, it's at the evolutionary biology scale, so very high diversity of shapes, super complicated to make it automatic. For human skulls, it's more, it could be closer once we have good landmarks, because all the steps could be time consuming. So that's what I wanted to show on this slide, is that everything that could save time on each of these steps are valuable. So segmentation to have a mesh, very valuable, or at least to have labels to identify key anatomical parts. Detection of landmarks, at least anatomical landmarks, and then further on, if we can have curves, that would be. But curves are more tricky. But once we have that, we could have very, very dense characterization. So automatic segmentations are also under development in lots of different labs. Also, automatic landmark detection is mainly done in orthodontic surgeons. So yeah, there is some research in every field, but it will take time until we have a very and also because there are shape repositories that become more and more available, that you can work on very huge databases, free access of faces, for example, of 3D scans, etc.
And
So there is a lot of data.
you have databases in the context of evolutionary biology.
Yes.
Okay. You know how they are called?
I can find them again. It's been a while. I find it again.
And maybe another question is that with Robin now, we have started really working with CT scans. And then actually you realize that, okay, bones are not solid objects, but actually, you know, they are semi-empty. Like you have huge variation in terms of the density of the bone tissue, which is something that we completely forget about when we turn them into surfaces, as if they were like big things. And I'm wondering, in your field, are people interested in those variation of density, etc.?
Yeah.
To
Yes.
abstract this away.
Yeah, absolutely. Okay. Yes.
Okay. So,
it's an important parameter. For example, in the facial bones, you know, there is pneumatization.
Pneumatization?
Yes. So it's the fact that it's there is a volume of air, and this volume of air is thought to be very important in the development of the bone and in the mechanical properties also of the bone. If you don't have that, you have you can have some a deterioration of the bone quality overall. And this bone density question is really important because it's telling us things about the mechanical properties of the bone, the health of the bone. So in the case of craniosynostosis, patients who have this kind of constraints due to suture closure, they have a brain that is pushing more strongly, and some patients have raised intracranial pressure due to this mechanical constraints. And if you have high intracranial pressure, basically you have this kind of patterns. I'm trying to go at the beginning. Yeah. You have this kind of patterns. You know?
You mean like holes in your skull?
Yeah, yes. So usually it's not really holes, there's still some membrane, you know, but yeah, a lot of skull defect, and so the quality of bone is really impacted because the pressure is too high. No.
When there were holes a bit everywhere.
No. It's really the consequence of the pressure exerted by the brain.
Okay.
Yeah.
Because it creates like, we're just looking at the surface, it creates like path from the outside and the inside. Like if you just take the distances between points, it can break everything essentially.
Yeah.
Because
this is real.
okay.
Yeah.
And so currently you still work on these as surfaces. So basically you apply a threshold, you extract the surface.
Yeah.
And basically, okay, because presumably what we would think would be to say, oh, you have to work with the image to keep track of these varying intensities.
Yes. So that's what we are. So there is one study we are actually mapping the bone density throughout the skull, but it's on the mouse experiment. Because the problem with clinical data is that it's retrospective data. So you cannot really control the parameters of scanning. So the Hounsfield unit, so the gray values in one scan will not necessarily mean the same density in another scan. So you can't really control that. So you can't really have a proper study on density based on retrospective CT scans. In this case, on the mouse, on one side we have settled the CT scan with the exact same parameters all the time for all the specimens, and also we included some phantom in the CT scan, you know, so that we have a real how to say, étalon, a scaling for the gray value with a real density estimate.
Why isn't this done in like routine CT scans? That doesn't seem like be something too hard to place an object there. It has a defined density, so you can like
for medical CT scans, you mean? Yeah, that
You
could be.
adjust. I mean, because it really seems like you place like a tiny cube there next to the patient, and then you know
Yeah.
like what the density is. And,
It could be nice, but again, if you even if we do it, it means that all the data for the past decades cannot be used. But yeah, definitely. But the radiology departments, they have like all the clinical departments, they have their inertia and habits. It's not so easy to change.
And just because I know that in rheumatology department, they have specific imaging devices where they can really look at the bone density and quality. And I wonder if you use that sometimes or never.
Not really. We have just started to be in touch with some people from nuclear medicine who are working with scintigraphy. I don't know if you heard of it. But it's you can inject some some small doses of radioactive substance that will attach to some for example a ligand to some proteins, let's say, that will be found in some places. So you can, for example, target bone formation. You can target growth cartilage, and so you can actually see where these isotopes are expressed, and so you can see, okay, this part of the skeleton is actually growing, this part is not growing, and it gives you clues. But the problem with this technique is that the resolution is quite bad. So it's really to have an overview of the whole skeleton. You cannot, at least for now, you cannot really use it to have a detailed image of a bone, for example, or the skull, or yeah. So I think I just had a couple of slides for a conclusion. So we need these shape analysis for many different reasons in clinical research, medical research. First, we need to assess lots of different things. So we need to assess shapes of the perioperative procedure in order to achieve the best aesthetics as possible. So basically, in the example of trigonocephaly, so the triangular forehead I was mentioning earlier, there is this whole puzzle where you need to cut the skulls at several places, and especially this part number 1 will be the new forehead. So this part number 1 here will go at the place of the front here. And so to achieve the best result in terms of aesthetics and also in terms of prediction on how the skull will grow with this configuration, it takes shape analysis, it takes mechanical model to predict how it will grow, etc. So it's really in the scope of what I was presenting. Oof. Okay. No, it's just little videos that are not working well. But also what we do a lot, and that's mainly my part, let's say, is the relationship between the form and the function. So what kind of function can you achieve with what form? For example, these patients with syndromic craniosynostosis, as I told you, they also have deformation of the upper cervical spine. So here there is the piece of the skull base with the foramen magnum, and this is first cervical vertebra, and this is second cervical vertebra. And so these patients, since they have very strong deformation of these bone pieces, they also have strong mobility and instability of the cervical spine. They can have a dislocation of the cervical spine that will produce some repetitive trauma on the spine, for example. So we need to better understand how the shape of these musculoskeletal system works together in order to achieve the right mobility. And because the surgeons, they will make some, they will modify the shape of the vertebrae. What we want to do is, okay, what should be the ideal shape of the vertebra in the end, so that we can find a good mobility. So that was also this the purpose of this study, to really find to understand the biomechanics and the kinematics of the upper cervical spine, and to relate it with the morphology. And so in the end, this shape information are used to be implemented in these parametric finite element models. So we have this standard model that we can arrange the geometry so that it fits any shape that we want, and then we can perform some mechanical tests, make some inferences on the forces at stake, etc. And this is just to illustrate that there are several teams working on these biomechanical models of skull growth in craniosynostosis. And the idea is that once we have a proper model that mimics the biomechanics of the bone, of the sutures, of the brain pushing, etc., then we can make some predictions on how one patient-specific morphology will eventually grow with these conditions. And then what we do is compare then the prediction with the actual statistical shape model, like the entire cohort, for example, so that we can evaluate the relevance and the accuracy of the prediction, and then we go back working on the biomechanical model until we achieve something really realistic. So that's the main application of shape analysis that we do in the lab. And I thank you for your attention.
Maybe a question is that a few weeks ago you showed us a very interesting picture of a bone that was
Yeah, I kept it.
and that changed topology in very unexpected ways. So
Yes.
Ouais, absolutely.
Yes. In terms of shape description for this bone, we will see that there is a very high variability.
can you just remind us, like, how is it called?
Ethmoid.
Ethmoid?
Ethmoid. So ethmoid is the bone that is just behind the nasal bones here. So it's the bone that forms the beginning of the orbital cavity here. And it's really the architecture is quite complex, and also it's all these, like the wings of the ethmoid are very, very thin. And so it's not really that the shape is chaotic in nature. It's just that we are not really able to make better reconstruction given the quality of the CT scans that we have. It's so thin and sometimes so poorly ossified that it's really, really difficult to reconstruct. And so once we have this reconstruction, it's very difficult to find good shape descriptors. So
the real
what?
bone is smoother.
Sorry?
The real bone does not really look like this. It's smoother.
Yes.
Not this.
So it's more like cartilage, which is ossifying.
Yes.
So it's around the thread.
Yes, and it's really thin. So given the voxel size of the CT scan, it will be difficult to reconstruct properly. Yeah, so it's kind of difficult. So I don't know if I had this. No. Yeah. So what we did is that we were able to find 3 on the sagittal crest and 4 on each side of the ethmoid, just to have this kind of dimension characterization of the ethmoid, not really the fine structures, and then we use thin plate spline so we have the best model, the best ethmoid possible that we have segmented, and then we deform it with the thin plate spline to make some interpolation of the shape between. But obviously it's not super, it's not perfect, but we can do better for now. And this one, so yeah, it started with this. So our the GM as I use it requires some manual intervention, especially for placing the curves, and it's really interesting if you want to delineate bone joinings. So all the sutures, for example, it's quite interesting to follow how these bones will eventually progress until it fulfills the anterior fontanelle, for example. So it gives us a good idea on how the sutures will close, etc. The idea is that sutures, even in most of syndromic cases, they are still visible even if they are closed. You can still see a little ridge. So you never have like mixing data in the landmarks. You're always able to sample the same landmarks on these sutures. But in other body parts, like the synchondrosis, it's not possible. So sutures are bone forming from membranous ossification. Synchondrosis is bone forming from endochondral ossification, so cartilage ossification. So here is the first cervical vertebra, and you see that at 1 year of age, the vertebra is looking like this. It's 3 different bone primordia ossifying from 3 different growth centers, and they progressively fuse together as ossification goes on. And the thing is, when the ossification is done, you cannot see at all where the synchondrosis was. So if I was willing to quantify statistically how this part is progressively fusing with the other ones, there's a limitation with the landmark-based approach. So what I did is, my students did, was to actually go use another method. I'm quite confused with my slides. So it's a template-based approach. It's very similar to the patch projection of semi-landmarks as earlier, but this time there is only one template, and you project everything on the target surface. So this is a template. It's like a standardized mesh with nicely symmetrized topology, and you rigidly and non-rigidly align this template to the target surface, and what you obtain is a new mesh that has the shape of the target mesh, but with the topology of the template. And so if you do that on every target surface, then you have a data set that is composed of the exact same number and topology of nodes. And so you can consider all these nodes as semi-landmarks. You can extract all the 3D coordinates and do the analysis later on. So that was the idea. And so we created a template of a vertebra and just introduced some topological differences by allowing the template to project on the target surface with topological differences. So there is one part that is more or less independent that will project on the small bony primordia on the anterior part, etc. So different topologies for the different configurations of the vertebra, and in the end we came up with this statistical trend of growth, where we could actually visualize how the synchondroses are progressively merging. So it was really bricolage, but it was a solution that worked. And statistically, we were also able to find back the real timings of ossification of the synchondroses.
Okay, because
It was quite satisfying in the end.
So because you're making approximations there, So obviously it's relevant if the approximation is smaller than the statistical thing
Yes.
that you're trying to measure.
Yeah.
And so what were you trying to measure exactly in this study?
So first is to have one statistical model that accounts for the whole little bony parts at once. And second, ideally, that the progressive fusion that we observe on this morphable model, it really corresponds to what we actually observe in terms of ossification rate and timing.
Okay. So you are not trying to correlate these times with a disease, but it's more like in a typical population, we don't really know at what age ossification appears, and so you are trying to document that.
Yes, so this is like the reference model, and then we also include different pathologies, to compare the rates and how and eventually how ossification at one site influences the growth at the other, in the rest of the shape.
So I have a question about the simplest splines. So every time you use it, this model, you are aligning points to corresponding points. So because I know this model, like you have to write a linear system and to invert it to find the optimal parameters. And that's what you do all the time, every time you use the model, or is it another, because since you are using landmarks, but also semi-landmarks, curves, and surfaces, I was wondering if you used in the end, it was always aligning points to points when you compute the simplest size, the TPS model, or you have something else? Or...
Yes, it's always correspondent. Yes, point to point.
Okay.
Yeah.
Okay, because okay, I think you.
Tell me. I'm interested, but yeah.
Because the simplest splines is really a model. It's like TDMM that we are used to work with. I mean, it's simpler actually than MDM, and you can use it with data attachment terms for points, but also curves, surfaces, and find an optimal matching for this energy of the simplest splines. So it will not be like just inverting linear system, which means you want to exactly align points to points, but something that would fit the points and then approximately the curves and approximately the surface. I don't know. It could be, but it could be. Right.
So concretely,
I mean, my understanding is that this is what you do with semi-landmarks. To me, it really looks, but we'll have to look at what is actually implemented. It really looks as a point-to-plane loss function instead of a point-to-point. It really feels like this. Like
I think it's even curves to curve and surface to surface. It's just that they place points on the curves.
Yeah. And
then they want to align, and
then
then they want to slide.
Yeah.
So that finally it's really aligning curves. And same for the surfaces. But there are data attachment terms that can really fit curves, not just points on the curves. And so
okay, so what you mean is like to ponderate different types of anatomical landmarks or semi-landmarks and give priority to anatomical landmarks and, let's say, curves, and not so much
like
to
you see the curve as a curve and not as a bunch of points. Because in your model, you are really aligning exactly the points on the curves, and then you let the points move along the curves, because in fact, you don't care about the points themselves, you just care about the curves.
Yes.
But there are approaches that really are able to match the curves independently on the points that are
describing the curve. So really using the spline and not the points themselves.
Yeah, it's based on points in the end because it's discretized, but there's no correspondence between the points. And you just.
Or maybe a way of seeing that is that basically you have your mesh, and instead of placing 10 points on the curve, you have a high resolution mesh. Use every single edge as a semi-landmark, or for surface, you would use every single triangle as a semi-landmark.
Yeah.
And so that's basically what this becomes.
Yeah.
Yeah, but it's also that there's no correspondence.
Yes.
Because so it's like
And so how would you label this a curve, for example?
Yeah, so
With the highest curvature detection, local curvature detection, for example, or
there are models this, but there is also basically like, for example, optimal transport based approach, or we've worked with Imagine even with currents varifolds, this type of stuff. So it's based on tangential information, and also it can be based also on curvature also. There is, so I think
this is definitely what we can talk. But yeah, I'd say, I think in medical imaging, we're used to thinking that we should have zero human interaction. So we're used to thinking that, okay, you don't want to rely on labeling. So I think the literature was more shaped by saying that, okay, you have like the whole surface is like one big patch with tons of small surveillance marks. And so what is a bit more original for me in what you present is that you actually have segmented the different pieces. And so I'd say in the medical literature, people used to think that this was too expensive, so we would not get access to this kind of mesoscopic level of information. But that's interesting. So in terms of design, it is very doable. And basically the whole story is that if you start to think that every triangle becomes a sub-million mark, then solving a simplex spline matching starts to become a bit expensive because it's not a linear system with 100 points that you have to solve, but you have more like 10,000. And that's why we invested so much time into GPU programming. That's how it.
Yeah, there is one guy from landmark-based GM. He has came up. I haven't checked the last updates, but he has came up with a set of libraries to actually do something like this. Consider all the vertices of a mesh as semi-landmarks. But he, I have to check again. But he like subsamples, some, not all the vertices because maybe it's 500,000 vertices and it's not possible. But then maybe the condition is that you need a good mesh, a mesh that is good enough to be really used fully, because I mean. One of the advantage I see in landmark is that you can still work. You see on the ethmoid, you can, even if I'm not really satisfied with the whole shape description, at least I could do something on the bone because I could find the 8 points. And in a lot of applications, it's enough. And it was not expensive or very heavy to do. So you can because the landmark, you really make a strong discretization, I think, of the information. You can work on deteriorated data.
I have maybe a question on why you're using landmarks. Like, are you very interested in the location of specific points? Like you've spoken a lot about homologous points, like on the edge between two bones and so on.
Yeah.
Are you more interested in these points than in semi-landmarks and other points when you're doing your analysis, or are you equally interested in all the points? And so you could maybe go into more like a fully automatic approach.
Yeah, it's a good question. I think it really depends on the question we have. If it's really to have overall information on how the skull vault is shaped, we don't really need homologous hypotheses, etc. But in general, in biology, we like to have homology because we want to know. Okay, there is a mutation. The whole growth, the whole shape is impacted. But now I want to know what bone, what part is more impacted, and how, if there is a compensatory growth at one side, I want to know exactly what bone or what amounts of bones surrounding a certain suture, for example, are impacted. So in this case, I really need information on what exactly is going on. If it's a rough geometry question, you don't really need this homology, I would say.
Okay.
In the previous figure that you showed with the regular mesh, this one, do you also use the method with the 3 types of lens?
No, this one we just use this approach.
And because when you explained this one to me, it seemed more straightforward, and I this one to do the
Yeah, good question. Okay, this one we so I opted for this method in this study because, you know, as I just answered, we did not really need a strong homology hypothesis. I just wanted to quantify the overall asymmetry and also the shape of the cheeks, because in this malformation, these patients, they have a strong asymmetry that is caused by muscular hypertrophy on one side, and it's really located in the cheek. So it's a place that you cannot place any landmarks reliably. So I opted for like a whole geometry face assessment. And so it's for faces, it's quite easy because it's a simple shape. It's more or less flat. There is no topological challenge. But for a skull, for a skull, you have orbits and you have cavities and foramens and stuff like this. The topology is really much more complex, and so it's kind of hard to use this whole template-based approach.
And yes, the surfaces are less nice here.
Yes, this one is super nice.
But still, you can kind of see in the object.
Yes, it's always a mess. And this is actually where the ethmoid bone is, you know? And it's a problem. Always a problem.
So you have to be a bit careful.
And also there is the inner side of the skull that is really complex, and the skull base. I don't know if I have an illustration of the skull base, but do I have one? No, I don't think I have. So yeah, the architecture is much more complex, and so using this kind of whole mesh approach is a bit more challenging.
And also in the same figure, you started by saying in this method there is only one template, as opposed to...
No. The other one also has only one template, yes.
I have another question about simple splines because, sorry.
Please go.
You use it to interpolate, to adjust all the landmarks, but then you don't use it for shape analysis itself, for the statistics. You go back to the...
No, actually, this is we use a thin plate spline to produce the where is it? Yeah, well, at least you know the bottom row, this deformation. So at the beginning of the presentation, there was a GIF with the growing skull. It's the same figure, but so thin plate splines. We mainly use them to produce some theoretical deformations along principal components, along partial least squares, regressions against different variables, etc. So that's what I was mentioning at the beginning of the presentation. advantage of this set of methods is that the answer to a statistical question is a shape or set of shape. So my question is, I don't know, what is the main statistical difference between two groups of patients? Let's say it's a combination of different principal components in a PCA. So if I display the TPS from one group to the other, then I have a deformation, a shape deformation, that tells me what are the main differences between the groups. So that's the main application of
the flow of
Yes.
of steps of your method.
Mhm.
And it looked like in the end you were not using here.
Could you say statistical analysis on the procrustes coordinate?
Yeah. So that
Yes.
I think that for us as mathematicians, we would say you should do the statistical analysis on the simplet splines using the simplet metric.
it's not that you should do, but it could be another option. Because
Instead of,
at this point you forget about the simplet splines that you
So
statistical analysis.
So thin plate spline would be used in the patching step, and in the end of the statistical analysis to produce the figures, to produce the visualization of the statistical. Yes.
With candle shape space.
With what?
Candle.
Yes.
Space.
Yes.
Okay. So it was never used. The TPS was never used for statistical analysis itself.
Directly.
Me personally, I've seen papers of people studying salmons.
What?
Salmons?
Salmons. And they were doing the statistical analysis in the simplet spline space.
Okay.
Not in landmark space.
Okay.
And for us, it seems cleaner to do it.
Yes.
So maybe they scan salmons and then.
Oh no, they take pictures of salmons and then they click. And then.
Oh, can you send me this?
Yeah, sure.
Because we go salmon fishing like every summer, so I mean. So I want to show the family.
So in concrete terms, at what point are they doing the?
It's more like when you perform your PCA.
Yes.
Because probably this is what you want to do. And the next shape seminar is someone who does a PCA on Neanderthal bones. 3 weeks, so come. It's going to be great.
Yeah.
And basically,
I will.
Okay, you want to do your PCA, and the question is, what are the input feature vectors that you give to your PCA thing? And one way is to use the landmarks up to rotation, scaling, and translation. And another way would be to give basically the vector that describes your thin plate spline deformation. And we kind of prefer it because if you feed to your PCA software the landmarks, then it means that if you have more landmarks somewhere, then it has more impact in the PCA. And say, oh no, we don't like that. So,
I don't know if it's better. I'm just saying that, for example, when you had this image of the sphere candle shape space with a tangent plane, you were saying that so in your tangent space, you have points and you do statistics on this in this plane. But this is with the candle shape space model. Actually, you could do the same with the simple splice model, I think. But it would not be. It would be directly on a tangent on a linear space actually. So maybe it's not the best in this regard because I don't know. Okay, so it's
yeah, but
I'm sure you have our packages to do both, and we are just curious about.
I just didn't hear about this possibility, but I mean it makes sense because when we are designing our landmark scheme at the beginning, we really make sure that the distribution of the ultimate shape descriptors, including all the landmark descriptors, will be homogeneously distributed on the surface, so we don't have an area that is oversampled and not the other one, because it leads to very well-known biases like Pinocchio effects and these kind of things. And so,
Yes.
Okay.
Sorry.
So it's a bias that is induced, especially in procluse superimposition. If you have not a very homogeneous distribution of landmarks, let's say you have a Pinocchio face like a square and the nose, and the nose is described by a point at the tip of the nose. And so the thing is, if only this point is moving, once you are aligning with procluse superimposition, all the points will move. And so if you sample some distant points, then you're increasing the Pinocchio effect, because the variance at this point will create artifact variance at the other points, although they are not moving. So we always pay attention to this homogeneous. But I would be curious, what do you think? What good aspect of doing working on the TPS directly rather than landmarks would bring?
It's exactly that basically you don't put more emphasis on parts of the shape where you have more landmarks. Basically, it's more like in terms of deformation of the space instead of.
But would not the TPS space inevitably be originally biased by the landmarks that you placed anyway?
No, I don't think so, because basically your deformation in the parts where you have many landmarks, okay, they are going to compromise. But in the parts where you have maybe one landmark, imagine you have many landmarks on the face, and one landmark in my right hand, one landmark in my left hand. Then basically, this landmark may be alone, but he has a big influence on basically what happens to the arm. And in the TPS space, the arm deformation has basically as much weight as the head deformation. So it's going to be basically same weight for the arm as for the head. Whereas in your landmark space, you would have a maybe 10-to-1 ratio, because the ratio would be.
Yeah, basically with the TPS space, you are really doing statistics on the deformation grids that you showed when you were doing TPS.
Basically, it's
not the landmark. I mean, in the end,
but still, how it will be interpolated between? If you don't have any landmark, for example, in between, then maybe it will not be at all realistic.
Yes. But I would say maybe the best intuition is to think that currently you do the TPS matching, and then you perform the statistical analysis, and you have basically you put importance weights by saying this is landmark. And what we propose, what we tend to use, is a kind of equivalent to saying, okay, I have my deformation, and now I throw away my landmarks, and to perform my statistical analysis, I put landmarks on the points on the grid. Like I resample uniformly, and this is what I feed to my PCA software.
And so
it's roughly that. So
and how would you place these new landmarks, ad hoc landmarks?
So basically, it's just that the formulas implicitly correspond to that, but you don't need to actually sample landmarks uniformly.
Okay.
But it's more equivalent to that. So basically, you put weights uniformly in space instead of putting weights where you have landmarks.
Okay. Okay.
But I have to say that's like your perspective. What we would do, I prefer this approach because I come from like the line of work. We took like Kendall's work and like extended like surface and elastic models. Right, to me, it seems like if you have a good matching, then usually like these things work super well. And
what works super well? The Kendall.
Kendall or the extensions to like surfaces, like these elastic shapes I'm working with, these triangle mesh deformations. Basically, like extension of the Kendall work, right? Kendall's like the first did this with just like point clouds, and then if you look at like triangle meshes, channel correspondence, then you get to our approaches. And so, yeah, the experience is if you have good matching, then they work super well in like deformations. I mean, I worked on Friday, but on Friday I showed like interpolation between different brain shapes, shapes of the brain between different species, and it looked super well. And they used exactly this approach, and they got like highly detailed, and which is like very tough to get, like thin plane sprites or LDMM or whatever. So
You mean it, because in your approach, you mean that you still maintain correspondences between.
Yes.
And you think it's better, give better results in the end. That's right.
I think once you have good correspondences, and it's typically easier with this intrinsic approach to get good deformation quality than it is to use anything that's extrinsic. I mean, also just by dimensionality considerations, right? Because I don't have to care about ambient space, which I don't need.
Yeah, but I would say here it's kind of an orthogonal question, because here the question is, once you have your deformation, how do you quantify big modes? And mathematically, it's basically about putting a metric actually on the deformations, and what you're doing, it's your metric is you have your landmarks, and so you make a sum. So like it's a L2 metric for your discrete measure, and instead
That's just clearly related to.
that you should use a uniform measure on your surface, be a better baseline at least. Okay. Just about weights, spatial weights.
But it's true, yeah, like Jean was explaining, if you have 10 landmarks here and just one here, and if the TPS finds that your deformation is like, for example, you expand a little bit both sides with the same, like symmetrically, then you will have a parameter for the TPS which is 10 times larger than the 10 parameters for the point here. And so you recover the balance between the fact that you have only 1 point here and 10 points here. Whereas for the crust analysis, you have the same weight. That's your idea, no? And I think that's true. In this way, it may be better to use it. Yes.
Okay. And is there any step of superimposition in the method you're thinking about?
What do you mean?
Like a proper superimposition? Is this something that you would still do? Or in the
so you can
if
do
you
the Procrustes alignment,
yeah.
no problem. And so basically it's really just about changing maybe the last line of your workflow, and it's really about saying how much weight you should put on the displacement on every point. And currently you put basically a weight of 1 for every landmark. And here instead you put a weight of 1 for every point in the space. So you weight things according to the space.
Okay.
Maybe what you would prefer is 1 point for every point in the surface.
Yeah, but we could do this more easily by just using like dual cells of the points and like actually assigning that proper like weights depending on the density, which is not too hard.
Yes,
It's just like nearest neighbor sampling and saying, okay, if I'm in a dense region, then I get a small weight. If I'm in a sparse region, then I get a high weight. And then this is a bit annoying to take a lot of points because you have like nearest neighbor, but it's just 3D. It's just a bunch of points. You do an off tree, whatever.
If you have a few points, it's even harder. Because
yeah, sure.
then it's like judging distance. If you have a lot of points, you don't care. You just do an 3 and it's for free. Yeah, it's a fight between intrinsic versus extrinsic.
It's not a fight. Just to tell you that basically when you do PCA, you want to pay attention to the weight that you put to your points, because it's easy when you have a table. You know, basically when you use PCA software with a table, implicitly every row has the same importance. And just that, you know, when you have a mesh, maybe you have large triangles, small triangles, and when you see them in the table, they all vote the same. Whereas maybe you want to re-weight, you want to say that the big triangle should have an importance, which is proportional to the weight.
Okay.
Basically the question.
So that's where you weight according to the closest neighbors, etc.
Yes. Okay. It's basically just a question of, basically maybe pay attention because it can be a bias in the studies.
Yeah. Do you have any idea, has this bias ever been quantified on real life objects?
I don't know, but you know, it would be easy to
yes. If you if we look together how to do this on DPS in the end instead of on the points, I can check what kind of variance it induces.
So basically, if it changes your conclusions, you think, oh, okay, maybe I have a problem.
No. I mean, I think we have also. It's interesting, but we also have different, how to say, concerns, and on biological real life work, sometimes we how to say, we agree to include some bias because it's sometimes a compromise between methodological challenge and what we want to achieve. So sometimes some biases are not super important to tackle, and so sometimes they are important. So that would be curious to see.
Okay, so very well. Thanks.
Thank you for this discussion.